And we saw that these cortices, they present a universal law. That's awesome. If you look at each of these objects here, all of them are folded. And we can use those variables to calculate, say, the thickness or average thickness, I must say, because thickness varies, but the average thickness, the exposed area, total area of each of these objects. And that's a way of expressing their shape. But how expressive is this expression? How much, by looking at those numbers, I can tell whether, say, this is a whale brain, this is a raccoon brain, and this is not a brain at all, that's a walnut. This is a bell pepper, this is a paper ball. Can I tell, looking at those numbers, whether I'm looking at a brain or a cortex or a non-cortex? Can I tell whether this is a human or a chimpanzee cortex? It's a healthy or sick cortex, if it's old and it's new? And the answer is you can, but in a somewhat convoluted way. It's not the most efficient way of expressing things. It's like when you write numbers in Roman numbers. I mean, I can always do this. Say M, C, X, Y, and say, okay, that's 2,111. Or I can do 2,111. This is saying the same thing, but this is far more expressive than this. That's what I mean by a syntax, a way of expressing things that is clarifying rather than complicating. And in this part, we're going to develop the syntax by both looking at the choice of variables, using variables that are more readily expressed, difference in shape and size, but also expressing things not in terms of single numbers, but in terms of functions. And that's going to enable us to differentiate between objects which are cortical, which are not, which are fractal, and which are not. And these came about by this epistemological, actually hermeneutical process of finding better and deeper questions. So let's recapitulate. I claim that the cortex crumples, that this is functionally behaving like this. But other people have claimed, and these people are not idiots, that the cortex actually warps, like when you leave a sheet of wood under the sun, or that it extrudes, like when you extrude pasta through a nozzle. And that also leads to gerification. That has been verified both in simulations and in actual physical objects that are growing in a vat of solvent that present these cortex-like structures. So there are many ways you can try to derive a mechanistic model for cortical gerification. And how can you compare these different models? How can you tell that my model is correct, but that guy's model is wrong, or his model is wrong, right, and mine is right? How can you tell them apart? The thing is that we start from the wrong question. Because instead of asking, how does the cortex fold, you actually have to have a much better understanding, a better description, a more discriminating description of what is the shape the cortex folds into. You have to specify the shape. We have to specify the shape better than we did before. So how we do it? What is the shape of the cortex? That's a more fundamental question in how the cortex folds. Once you derive, or you find a way of expressing an answer to this, once you find a syntax of describing folded shapes, so that cortex is a subclass of that class of shapes, then you can answer how the cortex folds in a much simpler way. So there are two alternatives that have investigated so far, at least implicitly. One is what I'm going to call the coarse-grained approach. You have this very complex object here, and remember, this is the cortex, this is not. This is the freaking cerebellum, the brainstem. But I'm just looking at this weird bit here. I take this whole object, and I express that in terms of a few numbers. A volume, an area, a thickness, a total area, an exposed area, and a thickness. There are many sets of numbers. We chose one that makes sense in terms of extrinsic, intrinsic size, and the size of the ruler inspired by our analysis of fractal dimensions. But any set of three variables, not any, but many sets of three variables could also work. Now, the problem with this approach is that the summary descriptions is uninformative, or it's not completely informative. I can, in principle, generate an object that has the same quantities of volume, area, and thickness, and don't look at all like a cortex. I can take a deflated football and contrive it to have the same values as my brain, and clearly that's not a cortex. So there's a limitation to this approach. Another approach is the one that implicitly we have been doing when we analyze the free surfer images or images from MRI. We said that we are investigating three nested surfaces. How we define the surfaces? We define the surfaces as triangulated surfaces. We have a set of vertexes, points. These are connected by edges, and sets of three edges envelop an area which are triangles, which are oriented areas. So a large set of vertexes, edges, and triangles gives you a shape of the cortex. And that, in principle, looks like much more promising because, okay, now I'm specifying a real shape. And this is not the same as the deflated ball or anything. But if I take this shape and rotate it, my sets of areas, edges, and vertexes are going to change. And although there are ways of verifying using representations of SO3 in a very clever way, that they represent the same objects, it's non-trivial. So this has the opposite problem, that a detailed spatial description is not very generalizable. I mean, if I have this, if I say this cortex is this massive set of 20,000 numbers, okay, this set of 20,000 numbers is a human. This set of 20,000 numbers is a chimpanzee. What's the difference? What's the difference between the 20,000 numbers of a healthy and a sick brain, or a brain of a 1-year-old and a 10-year-old? So, excessively detailed descriptions, they also have these advantages. So what we're looking for is something in between. Something that, well, I guess all you can read is local variables, global variables. We need something that is sufficiently detailed so that I can differentiate between various conditions and brains and whatnot, but also sufficiently generalizable so I can compare things and classify them. And that's what we're looking for, this happy middle here. And throughout this section of my talk, I'm going to slowly construct this happy middle, hopefully. So let's go back to what's the shape of the cortex. And I'm going to give you a very simple answer, but that's actually very revealing. Some are small, some are big. No? I mean, have small rodents, big cetaceans. This is an elephant, actually. Humans, this is a cetacean. This is a dolphin. Some are folded, some are smooth. Now, I said this before, but just to reiterate, if something is folded, we call gyrocephalic. If it's smooth, called leucocephalic. I might use both terms interchangeably, so that's what I mean when I do so. So what we're saying here, that we have to understand cortices at the most elementary level in terms of size and in terms of shape. Let's take two examples here. This is a cow, an actual cow. Gives milk, makes nice steaks. This is a sea cow, also a manatee. The Portuguese sailors call it a sea cow because it looks like a cow submerged in water. It also gives steaks and milk, but they're not as tasty. Now, they refer to animals that are about equally intelligent or equally stupid. They have brains that are about the same size. Both are mammals, but one is highly folded, almost as folded as us. One is almost as smooth. So clearly, size and shape, they are correlated. Bigger brains tend to be more folded, but that's not a deterministic relationship. In some ways, size and shape are independent. Not entirely independent, But what we're going to construct next is a set of variables, also 3 variables, that corresponds to a rotation in this 2-dimensional parametric space of our old variables, where we define 2 variables for shape and 1 variable for size, which are independent from one another. They are constructed to be orthogonal, and they're statistically independent. So how we do that? For that, we're going to study another system of objects, rectangles. So imagine you're a researcher and you found this species which you call rectangles. And you understand rectangles. So these are very rectangles that you see in the wild. The green rectangles, the blue rectangles. And you see that they can be characterized by 2 numbers, the height and the length. So each rectangle exists as a point in this 2-dimensional parametric space. But you see that some rectangles are exceptional. They have the same height as the same length, and we call them squares. And now you say, okay, so which triangles are more similar to one another? Which triangles are more different? Well, it depends on what you're trying to say. If I say I'm a farmer and I'm planting corn, then I'll say that 2 rectangular plots are the same, they have the same area. But if I'm trying to if I'm trying to make a drawing of a rectangle, also like rectangle, I'm saying that they're similar if they have the same aspect ratio. So we can define these quantities as functions of length and height. The area is the product of length and height. The ratio, the aspect ratio is the ratio between length and height. But by the magic of logarithms, instead of expressing these points in the linear variables, we express them in terms of the log variables, then things becomes very simple. So the log of the area is the sum of the logs of length and height. The log of the aspect ratio is the difference between the logs of length and height. Now suddenly, we can say that if you have an object of a certain area, this corresponds to direction, diagonal direction, so that all points along this line have the same area. If I have points that are along this direction, all points along this line have the same aspect ratio. In the special case of the square, they have this aspect ratio of 1. So what we have done here is that by defining these new variables, we define 2 different directions in this plane. This is same shape, this is same size. So we now segregate shape and size, and we can explain our rectangles for different purposes and different interests. So this seems very simple, but the idea that you can segregate shape and size is actually fairly general. And we're going to do the same thing with the cortex. So just note, I'm going to go over this again. The quantity that measures area is the diagonal between the primary diagonal between the 2 variables. In general, you can define an isometric size as the multidimensional diagonal of however many variables you have, as long as they have the same dimensionality. So in our case, we started from defining cortices as points in 3-dimensional space. Again, using the magic for logarithms, you have the log of exposed area, the log of total area, and the log of thickness. Now, there are 2 things you need to know. One is that this universal law here is obeyed. In terms of the log space, remember, power laws turn into linear relationships. Products of power laws turn into linear combinations. So this universal law here defines a plane in this space. And this plane can be defined as all the sets of points where this new variable k, or the log of k, is constant of a certain value. So we can start by saying that this k here is a new morphological variable. It carries information, combining information from all the 3 old variables, but it's an equally valid one. And the universal law then has a very simple interpretation. K is approximately constant. This 3-dimensional space can actually be reduced to a 2-dimensional manifold, which happens to be a plane. It didn't have to be, but happens to be a plane. So that's the first thing you need to know about K. The second thing you need to know about K is that it's dimensionless. What this means being dimensionless? Well, look at how it's expressed in terms of areas, always ratios between areas. I forgot to say, but instead of using thickness, I'm using thickness square so that all the variables have the same dimensionality. What does it mean? Well, imagine you have an area of 15, a thickness of 2. 2 what? Millimeters, meters, light years, doesn't matter. K is the same. So if I isometrically grow my object or shrink it, the K doesn't change. It doesn't know the size of this object. It's completely insensitive to the size of the object, right? Which means that we can find another variable that captures the size, and that's going to be orthogonal to it. And how we find this? Well, let's go back to the paper balls. If you look statistically, and again, this is one of you or two of you can actually do this. This is one of the projects I'm going to propose. You see, you're going to be able to see that these 2 balls connected by this purple line, they have roughly the same shape. They're just isometrically shrunken or isometrically scale up or down versions of each other. And this happens between these 2 and these 2 and these 3 and these 3. And what do they have in common? Well, what they have in common is that if I move from here to here, the thickness has changed, has doubled. The area, total area, has quadrupled, and the exposed area has quadrupled as well. Or in terms of these 3 variables that have the same dimensionality, the areas and the thickness square, all of them have increased by the same factor. So in terms of this 3-dimensional space, what does this mean? What this movement means? It means that you're moving along this diagonal. You're moving however many steps you want to move. Again, we're talking about logarithmic scale, so a step of a certain size is multiplied by a certain factor. So if I move along this direction so that I double this area, I move along this direction, I double this area, and I move along this direction, I double this area, I'm moving along this diagonal here. And if you look at the variables of log k, just look at exponents here. So the log of AT to power 1, the log of thickness, you have a power of 1 here, a thickness square, and minus 5/4 here, so the power of 1/4. And the exposed area to the power of minus 5, this is the direction of k. If I define this diagonal here as a sum of all 3 logs, it's very easy to see by doing the scalar product that these 2 are orthogonal. Another way of saying this is saying that if you have a vector that is, say, 1, 1, 1, 1, a scalar product between this vector and any other vector, that was called x0, x1, xn, is simply the sum of xn. So if the exponents of a vector sum up to 0, it's orthogonal to I. But if the exponents of a vector sum to 0, and these are logs of this quantity here, then this quantity is dimensionless. Because the dimension of it is going to be area to the power of the sum, to 0. So we have now 2 variables, one for shape, one for size, orthogonal to one another. All we need to do is do the dot product. We're going to have a 3rd variable. And this 3rd variable is going to be orthogonal to I, to the what I call isometric size, which means it's also going to be dimensionless. The sum of its exponents is going to be 0. And this is another variable for shape. Now remember, empirically, for courses, k is almost invariant. So you have shapes that change and become more complex or less complex. They can only move in a direction that's perpendicular to k, but in this shape plane composed by k and s. So if you increase s, you increase complexity. If you decrease s, you decrease complexity. We can call this s the morphological complexity of a shape. Wait, I think I skipped something here. So if you have, say, a bunch of points that obey the universal law, the more you move along this direction, the more complex you become, the more diversified you become, the more structures you have. You move along this direction, you become less complex. So we call this the morphological complexity variable. This is the universal law invariant, and this is the isometric size. KIS, keep it simple, that's how I call it. So this is a new set of variables that express each point in this parametric space, but now segregated in more usable and interpretable variables. I want to say just one more thing. The way you express this, these variables are not normalized. You can express them in normalized terms in terms of the numbers you get is simply the same values you had before to some power. If you express the normalized k, s, and I, expressing a point or a set of points, a set of cortices in the original variables or the new variables is simply a rotation. A rotation in three-dimensional space. You have a set, a base, you have another base. One is more convenient than the other depending on what the problem you want to solve. And that's really cool. We can actually take the data set for the comparative anatomy data for the whole species or for the human data, and we can do an analysis looking for how statistically correlated those quantities are. Obviously, since the universal law is obeyed, the original variables AT, AE, and T square are highly correlated. They have to be. But if I do a principal component analysis, I see there's three directions that corresponds to the axis of maximum variability, medium variability, and least variability. Well, we know that all the points sit roughly along the plane where k is constant. So k is very approximately the direction of minimum variability. That's one of the eigenvectors of the covariance matrix. And in fact, if you look at the set of directions given by the principal component analysis, they almost exactly coincide with these new variables that we have derived here. So what I'm saying is that these new variables that not only are interpretable in terms of shape and size, variance and invariance, but also they're statistically independent for the data sets we're working with. We can analyze them separately rather than have to analyze them jointly for the original variables. So these three new variables, we're going to be using them a lot in what follows. That's not a complete answer to our requirement of a new syntax, but that's a very good first step, and that's what has already applications. So we're not here yet, but we are this way. So we have still global variables, but they're independent. Now, what does this mean? Well, it means that we can now try to segregate different conditions for human cortices in this space. So we have three conditions here: temporal lobe epilepsy, aging, Alzheimer. Hopefully, in the next few months, some people are sitting here, and we're going to add more lines to this plot. And we look at initially for the original variables, the thicknesses and the areas. And what we see is that in all cases, aging, Alzheimer, and epilepsy corresponds to a significant lowering of the thickness. It seems, in fact, that every pathological condition in the brain, every degenerative condition in the brain leads to a thinning of the cortex. That's one of the most ubiquitous findings in neuroimaging. The areas, they change, but they don't change as significantly. So you could think that morphologically, all of these three processes, they are doing the same thing to the brain. But that's not so. The reason not so is that these three quantities are not independent. You cannot analyze them separately. If you look at approximately independent variables, you see something very, very, very different. So now look at the K, the I, and the S. Remember, the invariant shape, the morphological complexity, and the size. You see that the most important finding is that in all cases, the morphological complexity increases, so it's not very discriminating, but for aging and Alzheimer, the K decreases. And in fact, of all the variables analyzed, the decrease in K is the most statistically significant. But for epilepsy, the K increases. So using these new variables, we can now look at the variables separately. Shite. And you look at the K, you see that aging and Alzheimer, they look the same, similarly. Why? Because both involve the loss of axons. Both involve a white matter that's becoming less dense in terms of fraction of volume that's composed of axons. axovolumetric fraction. But for epilepsy, the opposite happens. For epilepsy, you seem to have more axons than you have in healthy brains. And why is that? Because you know that epilepsy, one of the causes of epilepsy is a lack of pruning of connections in the developing brain. You don't cut enough connections so that they have a brain that tends to be overstimulate itself, therefore leading to epileptic seizures. So what I'm going to show here, illustrate here, using this very simple example, is that these new variables, they're not only more interpretable, but they can actually discriminate between conditions that we know should be discriminated. And that's why I think they're more useful for the generating of biomarkers. Now, we can ask ourselves, these are all adults, some of them are old, but we're seeing that for these data sets, a snapshot in time for each data set, a data point. And you can ask yourself, okay, what happens between here and here? We know that the brain develops and then ages and changes in shape very significantly. The fetal brain starts smooth. It folds, it starts to become more folded as the baby is born, becomes bigger all the way up to adolescence, then starts to shrink. But this has never been plotted in one single plot because these are very different data sets. Until very recent, there was no baby data sets, and until very, very recent, there were no fetus data sets. But we have managed to create collaborations with people in India that have been able to extract the shape of the cortices for babies from, say, 15 weeks from gestation all the way to term. We can then combine that with the DHCP data for babies that are newborns. Some of them were born in between after 37 weeks, so they are term born. Some of them are preterm, they are born before 37 weeks. They're scanned between 1 and 7 weeks after birth. So this is the gestational week, and these color plots here, they correspond to the time when the baby was scanned. So very premature babies here, term babies here. And then we have a set of data points, or a set of data sets, different data banks about adult brains. Typically starting from adolescent, about here, that's about 15 years of age. And we can all plot them in the same plot. And I start with a simple measurement, the exposed area. That's a very good proxy for the size of the brain. And you see that the brain grows explosively in uterus, keeps growing through childhood and early adolescent, and then once you become an adult, it just stops growing. This looks like the typical case, the stereotypical case of you have development and you have aging. But we can do this for all the variables. Very briefly, we have the original variables, the total area, exposed area, and thickness. A number of things I can point out here, but I won't go into them because I don't have time. But essentially, the preterm babies, they seem to have a deficit of area when they're born. But for preterm babies that are not too early, they manage to, that cannot be seen here, but that's an analysis you can do. They're able to compensate for this deficit over the next few weeks after birth. But for a very early born babies, they never compensate. So it seems that very early preterm babies, they do have a permanent deficit, at least morphologically. Now we can also look at the new variables. I'm just looking at K here, and that's something really weird happens. You have this displacement here between the preterm and the healthy developing ones. If I zoom in only on the babies, I see this really cool picture where the brain starts, we know the brain has been growing. We've seen it grow in the previous plots, but it's not folding, just remain the same value of K. But after a certain period of time, then folding suddenly starts to increase. And then after birth, it keeps increasing. In fact, increases at the same rate. So birth doesn't seem to affect this very much. The process of brain developing continues before and after birth at essentially the same rate. And you can superimpose this with the very process happening in the brain. You have neural proliferation, neural migration, including tangential migration, myelination, synaptogenesis, where you start forming connections, and apoptosis, where cells are being killed. And you can, that's something we're doing now, but we haven't done it yet. We try to see if there's some synchronicity with these various aspects here. But one thing I can tell you, which supports our model, is that this increase more or less coincides to the projection and connection of axons. So it seems that the axon connectivity is necessary for the digitification to increase. And this becomes even more clear if you look at this quantity called the digitification index, the ratio between total and exposed area. So we see that it's very constant. In fact, it's slightly decreasing digitification index, and then it starts to increase again all the way to more or less the same rate as the term-born babies. So this is the sort of thing you can do with these new variables, with the old variables as well, with the right data sets. You can actually see how the shape of the brain changes before and after birth. Okay, I think am I early? Okay.
Once again, really nice talk.
Cheers.
I was thinking about naming. So, what are K, I, and S related to? Like, for it, I mean, it's easy to see the interpretation you're giving and how these variables can describe the morphology of the brain. But it does not have a name attached to it. It's just variables.
Well, I mean, I call them the invariant, the shape invariant, the morphological complexity, and the isometric size. Okay? So the isometric size is the easiest one to interpret. Because if imagine I have a shrinking ray, right? And I take these, I don't know, this cup here, and I just, here. I take this, and I shrink it, and it's this. Ta-da! Now, they have the same shape, they're just one bigger than the other. So if I plot these two in this two-dimensional space, these two are going to be lying along the I-axis. So move this is moving along the I direction. Okay? Now, the direction perpendicular to here is one that is insensitive to size. So this is where K and S are going to be existing. Now, K and S are choices. And these choices are driven by the fact of describing cortices. If I'm describing paper cups, K and S don't make any sense. I could choose any other pair of orthogonal variables in this plane. Now, we know that K is nearly invariant because of the universal law. So that's a natural choice, because that tells us what part of the shape development can affect, evolution can't affect. So S is a direction where evolution and development can operate. So that's the part of the shape that is affected over time. And we call this morphological complexity because more morphological complex brains have higher S, less morphological complex brains have lower S. So that's how we interpret these things.
Thank you very much for the talk. Could you go back the last slide? Yeah.
This one?
Yeah, the K1, the one before. Do you think the difference between fetal MRI scans and preterm scans could be due to the, as you argued for the cetaceans, the hydrostatic pressure of the amniotic fluid?
It's a good question. It didn't occur to me, but it might be. Do you know what the order of magnitude of this value of the difference between ambient and amniotic pressures? If they're significant difference at all?
Oh, I don't know. I'll need to check it because there is the hydrostatic pressure, the pressure of the uterus,
Yeah.
etc. So
Oh, look, that's a great idea. That's something you can check if someone has ever measured this, because that might be. Now, there's one caveat here to this data. Let's go over the full data set. So, like I said, measuring thickness means measuring the point where white matter ends and gray matter starts. And that's not always obvious. And in fact, if you measure the same brain using different scanners or different intensive magnetic fields or different protocols, the point where this boundary is defined changes. The same is true for exposed area to a lesser extent. So what I'm saying is that different data sets, even for the same population, or presumed population, can have systematic difference between them. So to get this somewhat nice line here, we have to do something called harmonization, where we have data sets that are for healthy humans with age and sex segregated, and we assume that they assemble from the same distribution. Once we assume that, we sort of stitch the various data sets together so that they coincide where they overlap. Now, this can be done between this data set and this data set, but there's too big of a gap to do this between this data set and this data set. So we have to trust that these things are roughly the same. But between these data sets of the babies and these data sets for the fetuses, we have harmonized between the term-born babies and the fetuses. Now, whether this is the right harmonization or not, I don't know. That's something we're still working on. But this big gap between these data sets and this, it can be true entirely, or it can be an artifact of the different scanning protocols. Because, I mean, in here we're scanning actual babies. Here we're scanning brains from aborted fetuses, right? No, I should have said that before. Yeah, these are actual fetuses. So just to the difference between live tissue and dead tissue is that the response of the relaxation of the spin, nuclear spin, is very different. So one we measure T1, the other we measure T2. So harmonizing these data sets is a non-trivial problem. It's still ongoing. Yeah?
So I understood the idea of K, S, and I is to have just orthogonal, like a
Yeah.
PCA kind of thing, but in a
Yeah.
informed PCA style. But in the example that you gave me, in all three conditions, before that's the one that you compared as, this one. Actually, the K, I, and S, all of them are different for all three conditions, right?
Yes.
So here, actually, in reality, you do you have any example where actually you see a difference in one direction, but not in the other, or this kind of thing?
No. I haven't seen any conditions that have this. I mean, one thing you can do, that's something we haven't used this yet, but one thing you can do is to actually find the linear combination of the logs of these quantities that corresponds to the maximum variability of, say, aging of Alzheimer, or even sex, if you want. And we can say that that's the direction that changes the most. But we have analyzed this for these conditions and some others, and I don't have a clear interpretation of what these new variables mean, except like circular definitions of these is the measure of Alzheimer-ness of a patient. But these new directions can be used as biomarkers. They are not great biomarkers, but they're better biomarkers than we have for single coarse grain variables. But yeah, that would be cool.
Thank you.
Okay? Okay, so what I'm going to talk next is, well, one of the coolest things I think we have done, which is the melting of brains. Wait, here. But also, one thing that shows the limitations of the things we currently do. So I'm going to talk about the brain melting, and then talk about the fractal brains that we can show they're fractal because of the melting. But then I'm going to go into something that is really not done. That's something we're currently working on. We don't have an implementation yet. But it's another way of expressing self-avoiding surfaces, not as mapping between two-dimensional and three-dimensional spaces, but rather as equipotentials in a scalar field. That's, I think, one of the most promising approaches for shape analysis. And that really, really enables us to express a shape in terms of orthogonal components evolving through time. That's really cool. But like I said, this is not even preliminary data. That's work in progress. But I think it's a nice way to finish this with what has been going on in my lab. So let's start with the question. We said that progress in this line of research is really about finding better questions to ask. And we move from how does the cortex folds to what is the shape of the cortex. But the next step is a bit more important. It's a bit more subtle. But it's think of things in terms of statistical physics, in terms of ensembles. See, not what the shape of one cortex, but what are the shapes of cortices. So what's the difference between those two questions? What's the difference between asking what's the shape of the cortex, what's the shapes of cortices? Is that you want to quantify, want to specify the ensemble of potential folded shapes that can correspond to cortices. Real cortices or imaginary cortices, but there is a subclass of folded shapes that we can define that corresponds to cortices. So that's what we're going to ask. Among all possible unified structures, which may correspond to cortices. So we want to define this subset. How we do that? Well, we look for the universal law as a starting point. So as we discussed before, this is a relationship between the dimensionless measure of intrinsic size and a dimensionless measure of extrinsic size. So this looks very much like the standard law you obtained from the scaling of approximants of fractals. Doesn't prove that the cortex is a fractal. Remember, this was obtained not by analyzing single cortices over many scales, but analyzing different cortices. So this is just suggestive. But if this is a relationship that is due to fractality, remember, sort of relationship here, intrinsic size divided by the size of your ruler, extrinsic divided by the size of the ruler, the fractal dimension to some power. If this is a relation derived by the fractal dimension, then this exponent here must be the fractal dimension divided by 2. Why divided by 2? Because these relate areas, right? So this exponent here is going to be the fractal dimension divided by the topological dimension of these tiles. So if our cortices, they have this scaling law because of fractality, the fractal dimension should be 2.5. Well, empirically, a group measured the fractal dimension of a cortex using a box counting algorithm that is slightly improved by making the box shrink and grow in clever ways. And they found the fractal dimension 2.49. This is really cool, right? I mean, it suggests that we may be on the right track. Now, this is really good, but that's not sufficient because, like I said, we define the cortex as a set of 3 nested surfaces that are both self-avoiding, they cannot intercept themselves, and mutually avoiding, they cannot intercept each other. So once you start to take your voxels and growing them, you start breaking that condition. So something that looks like, say, a deflated ball that you crumple in weird ways, starts looking like a sponge. You no longer are able to measure things like thicknesses and areas the way we did before, or K and S and I. So these approximants here, they're good enough to calculate the fractal dimension, but not to do an analysis to prove that this thing is actually a fractal and what kind of fractal it is. To do this, we need to do an algorithm that do a coarse graining, thus eliminate, so look at this object at low resolution, but while preserving the topological geometric integrity of the surfaces, something like we're doing in this poor dog here. And we have devised such an algorithm. The algorithm, it moves back and forth between a voxel-based approach and a surface-based approach by hacking some algorithms, some algorithms that free-surfer uses. So we start from a segmentation, we say gray matter and outside, or gray matter and white matter, and use the free-surfer algorithm to delineate the surface between those two. Now we do a coarse graining. So each, say, 4 voxels become 1 voxel. If this reminds you of renormalization, yeah, that's the initial inspiration. In a way, it is a sort of morphological, empirical renormalization. So now we have these big voxels, and now this is the key. We need to do a new segmentation. And we use the original surface to see whether the voxel is inside or outside. If it's inside, then it's gray matter. If it's outside, it's the new outside. Now, we can use the algorithm that we had before to generate a new surface. So in principle, there are some complications with the fact that cortical surfaces are really, really complex. But in principle, broad strokes, that's how his algorithm works. We start from one surface, we generate a new surface, and then a new surface. So these new surfaces, they have this property of this approximately, because this method is not perfect, they generate surfaces that are self-avoiding, mutually avoiding, but also, crucially, they are surfaces that have been coarse-grained so that details smaller than the typical size of the voxel become erased. I'm erasing the smallest sulci and smallest gyri, or sulci and gyra is smaller than the voxel length. And when you do this, this happens. This is the gray matter, this is the white matter. I apply this and make a movie so that each frame, the size of my voxel increases. The threshold, the size threshold beyond which I delete structures increases, and that's what I get. The cortex melts. It becomes less folded, all the way to lissencephaly, and proportionally thicker. So this is something that looks like a rat brain or rabbit's brain. But of course, we have to quantify this. And we have done this for 11 different primates. I need to do that primates because that's the only way I can make free-surfer work. We have now melted the brains of a sea lion. We currently melted the brain of a dolphin. They also obey similar laws, but that's what we get. It's less precise than the human because the scan is not so good and the free-surfer algorithm is not so good. Now, we can do the same for things which are not cortices, like a walnut, because some Yahoo just decided to put a walnut in an MRI machine and leave the scan on the internet. And have something that qualitatively looks the same, but as we're going to see, quantitatively, it does not. It's a walnut melting. Okay, so what do we do with this? Well, each frame of this, because we preserved geometry and topology, has a total area and exposed area of a thickness. So we can express these movies, each of these movies, as a trajectory in the KSI or AT, AE, and T square space. Is that what we do? And then something really amazing happens. Because we start from this very species. Each species corresponds to a different color. It started at one point in this two-dimensional space where we first obtained the universal law. And as we increase the voxel size, we plot the new shapes like this. These are humans, these are chimpanzees, and various other monkeys. And we see that not only have straight lines, but we have straight lines that coincide. They follow the same track. If you look at the slopes of these lines, they almost perfectly cluster around 1.25. Each of these points is one specimen. Each color is one species. So humans, chimpanzees, marmosets, everyone folds the same. Now, this is a more powerful statement than just saying the universal law is obeyed, because I'm saying that it's obeyed over all length scales. And this proves that the cortex is a fractal. How so? Well, because the fact that these lines are straight means that these are self-similar objects. That a melted cortex of a human looks like the big cortex of a human with the details removed by the same scaling laws. And that a melted cortex of a human looks like a chimpanzee cortex. And that a melted cortex of a human chimpanzee, the proper scale, look like a baboon cortex. So what we're saying here is that each one of those movies of the melting brains separately obey the universal law, which can express as this: a relation between total area and exposed area measured in units of the fundamental size. And the slope is almost exactly 2.5. So let's step back and think, okay, what does it mean when I say something is a fractal? Because fractal are mathematical abstractions. Fractals are things that are infinitely detailed. Fractals are things which don't have smooth edges at all. We're saying that a tree or a cloud or a brain is a fractal. We're actually saying that it's approximately a fractal. So if they all follow the same trajectory, what is the difference between my brain and a giraffe's brain and a chimpanzee brain and an Alzheimer's patient, not Alzheimer's patient's brain, but a marmoset brain? The difference is that, I should have said this before, but I can do, is that it approximates, each one approximates a fractal, but not only approximates a fractal, approximates the same archetypical fractal. The fractal is whatever lies infinitely along this direction, where I have an infinitely large brain that's infinitely girified. So they are approximately the same cortex. So what's the difference? The difference is the range of sizes for which the approximation is valid. So for human, you have, say, the exposed area. It ranges from here, the actual exposed area, to here, the point where the brain becomes encephalic. For the chimpanzee, the range is different. For different types of primates, you have different ranges, and that's the main difference. That's the main morphological difference between my brain and that of any other primate, and presumably any other mammal, healthy adult mammal. Right? That is, I think, the key to shape. Right? We all fractal. We all approximate a fractal. How well we approximate a fractal differentiates our brains from our neighbors. Now, this can be quantified in a way that I won't go into detail here, but these accounts for most large-scale differences in morphology. I think that's a profound statement, and I think that's something I've actually been able to prove rather than suggest. Now, just to see how we quantify this, remember we defined a quantity called the girification index, the ratio between exposed area and total area. Well, if this indeed follows this universal law, then we can see that the ratio between the largest structure, which is the whole hemisphere, and the smallest structure, which say 0, the ratio is a power of the girification index. It's a calculation that can be easily done, but I won't do here, is the girification index to the 4th power. And the same is true for every other variable that we derive from the original 3 variables. So if we express the thickness, the exposed area, the volume, the gray matter volume, the number of features, how many substructures morphologically one, the K, the S and I, the more normalized K, S and I, we have the original value, we have the span, which is always a power of the girification index. So what I'm saying here is that nature doesn't have many degrees of freedom to fold the brain. All the variables are tightly correlated in terms of the range they can span by the girification index. And all that nature has to choose in terms of these, at least in terms of the dimensionless quantities, is the range between the smallest and the largest structure, which I think is an important thing to say. Evolution and development can work elsewhere, but not here. This is physics. Now, just to finalize this analysis of melting, I want to first express this in terms of these new variables to show how much easier it is to look at that. So this is expressed in terms of the original variables, the one exposed area, the one involving the total area, the thickness. That's like from the model, the entropy component and the energy component. But if you look at the new variables, the ones that we know are more interpretable, we can look at this in terms of the dimensionless variables. And what you see is that, like I said before, this is the data for humans, adult humans, and we start melting one of these humans along this direction. And the gray points are the compared anatomy data for different species. And you see that essentially all of these lie along the same trajectory. There's some discrepancy here, but the difference is actually very small. If you look at the actual values that are changing here, we talk about tenths of units here, as opposed to entire units here. So what you see is that as one melts, we move horizontally, we decrease morphological complexity all the way to lissencephaly, that's indicated by this line here, while maintaining an approximately constant K. This is how we express a shape as a trajectory. And all these trajectories overlap the same universal trajectory. And to see how this thing could be different, let's imagine this in a more abstract way. Imagine you have one individual, can be you. So it has a certain value of k, certain value of s. Now, we know that this individual has to be Girified because this line here corresponds to brains with a smooth. Has a Girification index of 1, a fractal dimension of 2. Now, if I melt this brain, I have a trajectory that goes like this. It means that this is a fractal trajectory because it has a straight line. It has a fractal dimension of 2.5 because in this case the trajectory is flat. Now, in principle, if the brains were not obeying universal law, we could have something like this. This is also fractal, but has a different fractal dimension. Or something like this, that's not fractal at all. Or something like this, has the same fractal dimension, but it's a different fractal, approximates a different fractal. But what we have is this. Different species follow the same line, they just have a different span, but they all have the same fractal. And finally, in terms of the Girification index, since the Girification index is also dimensional, it's a ratio between areas, it also corresponds to the direction in this plane. So we can find these various contour lines to various values of the Girification index. And this is the Girification index of the original brain. And as you melt it, it decreases, 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 so that we have this span between the actual Girification index and 1, and that corresponds to the span of S and the span of AE, AT, and every other variable that we have. So that's what I mean by universality. So this has been fairly recently published. It took like a long time to publish this thing. It fell into this uncanny valley between too theoretical for the biology and too biological for the theoreticians. But I'm really proud of this work. I think this says something really deep and universal about shapes in codices. Okay, now I want to summarize what I have found. We found that describing a brain, a cortex, in terms of a whole load of numbers, it's not very generalizable. Defined them by very few numbers is not very descriptive. It gets a little better if you use independent variables, but the really, really big step forward is to express this shape in abstract in this three-dimensional space as a trajectory. Comparing different trajectories can allow us to better differentiate between youth and old age, health and disease, and different species. And that's something that's actually doing right now. Just to give 2 examples. If you look at, first. So what I have proven is that there are actually 4 aspects of the same universal law. It's a value across species, individuals, cortical regions, and now length scales. And let's now apply this last finding to better differentiate in 2 cases, aging and Alzheimer. So why do we melt? What's the use of melting? So what you do here, I had a caption that's somehow misplaced. So what we have here is 2 different sets of brains. Adult men of 20 years of age, adult men of 80 years of age. So adult 20 years of age red, 80 years of age is blue. And you see something really cool. We can measure the K as a function of the scale. So if, say, the scale is 4 millimeters, means that we raise every sulca and gyre is more than 4 millimeters. Here we show the statistical significance of that. And this is an illustration of what actually looking like for the 80-year-old and 20-year-old. So what you see is that if you are young, 20 years old, the K just decreases monotonically all the way up to, say, about 2 to 4 millimeters, then increases slightly. So the point where the K is maximum is actually the highest resolution. But for older brains, a K actually increases up to a peak of about 2 to 3 millimeters and then decreases. So clearly, the ratio between the values of K shifts as one changes scales. So this carries more information than the K for the original value itself. And this can be quantified. So this is not a great biomarker yet, but it shows that there is information across scales. And we use area rather than K because it's easy to interpret what area means in terms of scales. You're just agglutinating more area as you add more and more sulci. So the green plots here are the variance of our estimators for age using only one of those scales. So if you use the native scale, you have a very poor discrimination, about 20-something years of age. If I melt the brain down to 1.85 millimeters, which is about the peak here of this blue line, I have a slightly better discriminator, so the variance of 18 years of age. It means that if I take a brain, I scan a brain, and I use this marker here, I can guess the age, the plus or minus 18 years of age. But if I combine those two information from both scales, I can decrease this uncertainty to about 12 years of age. So I mean, it's not yet a biomarker that I can use for a general application, but it shows the promise of this approach. By extracting information from many scales, you can actually differentiate between conditions that are very similar. So that's an application for aging. We kind of understand what's going on, but we can also do this for Alzheimer's. That's actually very cool. So in here, we have plotted the same value of K as one melts the brain, and that's the average for healthy humans. Now, we have some patients in Rio that I scan every 2 years or so. And some of these, they're given a battery of cognitive tests. And then they're classified as healthy, mild cognitive impaired, or Alzheimer. And in some of these patients, they have a transition between mild cognitive impairment and Alzheimer. We see something that I think is really cool. So the mild cognitive impairment patients, you have approximately the same trajectory, except the largest scales. This may be an artifact, we don't know. But once you develop Alzheimer, we know that K decreases, but we now can see at which scales that K decreases. And you see that it starts to decrease at smaller scales, and it's like a zipper, it starts opening up to higher and higher scales. This is also very recent. This is still under work. This is actually, it's actually under embargo in a way. So, I mean, I had to ask authorization to show it to you. But this shows you how these methods of comparing not single variables, but trajectories can actually hold a lot of promise for biomarkers, precise biomarkers for various conditions. Okay, so I now have introduced everything I want to introduce in terms of the methods that we have used. So now I want to step back a bit and talk about the limitations of these methods. We talk about the brain as, or the cortex, as having 3 nested surfaces. And in fact, that's our geometric description of this cortex. But that's not all. The surfaces, they're closed, or at least can be closed. They are simply connected, so they don't have handles, and they are self-mutually avoiding. And the problem is that once you start reconstructing surfaces from voxels, from MRIs that have lots of sources of errors and imprecisions, all of these three go out of the window. You have to reinforce these conditions after you generate the images. That generates a lot of artifacts and a lot of grief. So what if there's a way or generate surfaces that come with these qualities out of the box? And that's what I'm suggesting here. I want to generate surfaces that have self-avoidance baked in. And the way you do that is not describing them as mappings of, say, either a sphere or a plane into the 3-dimensional space in some complicated form, but as equipotentials of some scalar field. If you imagine that the volume is filled by a scalar field that assumes a real value at each point, you can find a surface that corresponds. If this field is smoothly varying across this volume, we can find the point that corresponds to a certain value of this field. And these surfaces are going to be extremely well behaved. If we can somehow code our surfaces as these equipotentials, this might solve all of the problems I described before and many others that are still to arise. So how do we do this? Imagine, this is an analogy, I'm not putting brains in microwaves yet, but just imagine to have the white and gray matter boundary, and this shape has a certain temperature, let's say 100 degrees, which units, I don't care. And then the pile surface I put in contact with a thermal reservoir that has 0 degrees. Whatever the initial distribution of heat, of temperature inside this gray matter, assuming that the gray matter is a perfect conductor that's perfectly homogeneous or whatnot, eventually, I can let this system evolve. Eventually, I'm going to reach a stationary state. Heat's going to flow from hot to cold, but the temperature at each point is going to be a constant. So when I do this, I can say, okay, this red is an equipotential. That's my counter conditions. Equipotential for 100 degrees. This blue is the equipotential for 0 degrees. In between, I'm going to have equipotential for 75, 50, 25, whatever intermediate temperature I want. And then if I take the gradient of this scalar field, defined by this temperature field, well, it's going to be perpendicular to each one of these surfaces. So I now have a sort of coordinate system where I can have a mapping between the two surfaces, and these curves, in a way, give me a measure of thickness, and I can have, orthogonal to that, a bunch of surfaces that I know are going to be self-avoiding, and they have this, assuming that the red and blue surfaces have the same, they are homeomorphic to one another, they're homeomorphic to a sphere, they're also going to be homeomorphic to spheres, and they're going to have no saddle points. Everything is going to be perfect. Assume I find a decent enough a scalar field, but I know how to find this scalar field. Because heat in this ideal condition is described by the heat equation. The temperature at a certain point changes proportional to essentially how much energy comes in minus how much energy comes out. It's Laplacian. Now, if I look for the stationary distribution, that's a distribution where the temperature at each point is constant. So this equals 0, and that is Laplace equations. It is literally the most well-behaved equation in differential geometry. So if I just define these counter conditions here, I solve the field for these conditions, I have a set of equipotentials that are extremely well-behaved. And that's what you want to do to obtain the equithermals and the gradients. So that's the basic approach, what I'm going to tell in the next 20 minutes or so. So this is what you do. This is a really simple Python code in 2 dimensions. Every example I'm going to give you is going to be in 2 dimensions. We're implementing these in 2 dimensions. It's horribly complicated for actual brains, for reasons I won't go into. But the idea is very simple. We have the hot contour, we have the cold contour, and in between, we have the intermediate contours and the gradients. Now, if you integrate these gradient lines, you can think of this as a generalized measure of thickness. How far is the surface from this point here? Well, you can take the straight line, but you cannot do that in a self-consistent manner for all points. You're going to have intersections, have squeezes and stretches. But if you think of this thickness as the arc length of this curve from here to here, then you have a very well-behaved thickness, and one that's univocal, because if you propagate back, you have the same trajectory. So we actually done this for brains. This is a slice of a 3-dimensional map, and we can actually show that the thickness distribution is much more diverse using this, what I call Laplacian thickness, than just using the thickness that free surface calculates. And free surface calculates thickness in a way that's horrifying. If you know anything about geometry, you're going to be horrified by how it does it. Because it does is it takes 2 surfaces. And then it finds this point here. Okay, what's the thickness at this point? It finds the closest point by a freaking straight line to here. And then it finds the closest points to this point back to the original surface. And then takes the average. Does this make any geometrical sense? Please say no, because that's, it works approximately, but it's nonsense. It doesn't mean geometrically anything, at least a very rough approximation. But this does. And what we show is not only that you can find much more variety, much more dispersion in the thicknesses of the cortices, but they do so in a way that makes sense. Different regions of the cortices are thicker than the others. We know that that's true. And we can actually map this and make column maps of that. I want to go into this because that's, again, another whole line of research, but I'm just saying that these Laplacian methods by themselves allow you to measure thickness in a much more precise way. There's also a very intriguing possibility, because so far we have been discussing gray matter and white matter as these amorphous blobs of homogeneous matter, but you know that's not true. White matter is composed of really crisscrossing axons interspeed by oligodendrocytes that myelinated them. And all sorts of things are happening there. Gray matter is composed by these very complex neurons that have projections everywhere, but they are structured as they have developed and migrated in cortical columns. So neurons, they are generated by a progenitor cell, and they stack up, they move vertically, and form the cortex as piles of sister cells. And these piles of sister cells are what we call cortical columns. And if you look at a brain in a microscope, you see that it is formed by layers that are very easy to discern, 6 layers for our cortex. So what we have here is a structure that has a radial component and a tangential surface component. So one thing that Laplacian method can do, and that's something we really want to do with the babies from India, is to see whether the gradients correspond to the direction of the cortical columns, and to see whether the ecthermals corresponds to the layers of the cortex. And the answer is going to be interesting whether that's true or not, because if it is true, it's indication that the cortex is in some way in equilibrium, and that the development process in a way mimics the flow of the heat in our model. If they do not, there may be indications of out of equilibrium systems, so that might be something we observe in developing brains. So that's one thing we can do with this Laplacian method, but there is more. I mean, it's like these Ginsu knife advertisements. We need decent white matter and gray matter surface, and that's actually the hard part. That's something we're still working on. But there's more. Because we talk about the stationary distribution of temperature, but we can do more with non-stationary distributions. If you look at the heat equation, one way of solving that is looking for a station. It's so nice I can say this to you. Sometimes I'm giving talk to, say, medical students, people who don't know much math, and I have to really work hard to explain what all this means. So if you have the temperature field, it's a map, it's a function of position and time. But you can imagine that is the product of a time function and a spatial function. And then this equation becomes separable, and the time function can be easily solved, and you show that when you do the separation, there is this constant lambda that has to be positive, and the time component essentially is a exponential decay at rate lambda from the original value. And the spatial component is a solution of Helmholtz equation. So this is the general way of solving an arbitrary distribution of heat in space. And when you do this, you find that this is, there are a number of functions that obey this equation, each one associated with one different value, which is an eigenvalue. So the general solution for the temperature distribution evolving through time is the initial distribution of these various I'm going to call eigenshapes, or eigenmodes, each one decay at a certain rate. And there's one specific eigenvalue that's always there, which is 0. So the 0 eigenvalue does not decay. And that's the solution of the Laplace equation. So when you have an initial distribution of heat, as it evolves through time, all the modes that's not the 0 eigenvalue mode, they decay, and what's left is the stationary distribution. And this can be used to generate melting in a way that's much more elegant than I have done before, like this. So I, again, create a gerified surface, and I started to evolve it through time, and what you see is that it evolves, the sulci become less pronounced, and the cortex becomes more smooth. How does that work? Well, since I call these fading modes of heat, and it reminded me of like a progressive rock albums, I actually asked ChatGPT to produce it. I really want to listen to this, but it doesn't produce music yet, just the images. So what those these fading modes of heat are? They are these eigen shapes. Initially, for this star shape, you have only one boundary. You have to define a temperature for this boundary, but that's trivial. And you see that these various eigen modes corresponds to geometrically sensible things like a radial component and a dipolar component. But what happens is as we evolve through time, the higher the eigenvalue, the faster these components decay. So you see that as we evolve through time, the higher all the components, they decay faster than low other components. Please. And we can use that to do melting. Because if we have 4 surfaces, the original pile and white matter surfaces, and we have the convex hull on the outside and something like an inner convex hull on the inside, we can define a Helmholtzian process where the surface of the thin blue line flows to the thick blue line, the thin red line flows to the thick red line, in a way that we can associate with a certain scale by the heat equation, and that we can decompose into components. So each component corresponds to melting at a different scale, given by the inverse of this lambda here, or the square root of that. So we haven't done this yet, because in 2 dimensions that's a very hard problem, but that's another way of melting. I think it avoids those issues with the artifacts that our current algorithm generates, and many, many other things. One of which is that we can actually reverse time, so we can melt in reverse. And finally, I swear there's the last thing I'm going to tell you about. If you look at these components, typically you have a radial component, and then you have 2 dipolar components, which means temperature distributions that are hot in one direction and cold in another direction. That's very ubiquitous. If you have a situation where you have more complex shapes, they still appear. So here are the dipolar components of these fractal shapes. And you have the radial component. And you have something that is on a sphere. Who knows what is this? What is this? Any astrophysicist here? What? Say it loud. What is this? No, say it. What is this? Yes, CMB. What is CMB? Is the after you remove the dipolar component, is the fluctuations in the temperature of the cosmic microwave background. So you can think of this as temperature defined in a sphere. And if you imagine a sphere that has this pattern of temperatures here, you can see what happens when it evolves. What happens as it evolves to time? Well, eventually it's going to evolve to a homogeneous temperature. But before it does that, there's some interesting things. And when you do that, then we can find that is that we can decompose any such distribution in terms of the multiple components. And these multiple components are given by the order of this value of L. So the eigenvalue are going to be given by this product here. If you study quantum mechanics, you've seen these things before. Because, I mean, SO3, SU2, whatever, blah, blah. But the thing is, for the first mode, you have only one component. And that's the monopole component. It doesn't say anything about where you are. If you imagine the temperature of the monopole component, you're going to be the same temperature at any point. But then you have 3 components that are like, if for someone in Earth, you don't know where, and it's hot, it's more likely near the equator. If it's cold, it's more likely that near the poles. Now, this generates 3 sets of poles, so that it's cold up, hot down, cold in, hot out, cold right, hot left. So if you measure one of these components, you measure the temperature, you know where you are along one of these axes. And then you have the quadrupole, octupole, and other modes that are more detailed descriptions of the temperature distribution. What I'm saying here is that if I decompose this temperature distribution into these first modes here, I can actually say where I am. And that's exact. If I look at the descriptions of these monopoles, the functions for these monopoles, if you give you the value of these monopoles, you can reconstruct X, Y, and Z. Like this. You tell where you are in X-axis, Y-axis, and Z-axis. That's very simple to do. So that's what you get. Each of these spherical harmonics gives X, Y, and Z. Now, why I'm telling you this? Because no one is melting spheres. But if you do the same thing for a surface of a cortex, if you decompose the Laplace-Beltrami operator on the surface of the cortex, you always find the same structure. One monopolar mode that tells you nothing, 2 dipolar modes that tell you where you are along axis, along this object. And in fact, if you talk about courses, typically these axes are mediolateral, dorsal-ventral, and rostrocaudal. So they correspond to the general developmental axis that we obtain in the cortex. But there's more, because we can do this for all sorts of shapes and obtain all these modes of heat. And this allows us to do something called spectral embedding. Initially for surfaces, but again, there is more. Because if you now think about hollow objects, now we can do the same thing we did before. We have the dipolar mode, we have a radial mode, but if I impose a condition that the inner ring is colder than the outer ring, now the monopolar mode is no longer trivial. It's a smooth mapping between the hot and the cold. So now it gives you a radial coordinate. And what you get is, in 2 dimensions, one component gives you the radial coordinate, and 2 components, dipolar coordinates, that give you the x and y. And in the end, I swear I'm finishing. This is the solution of the Laplace equation. This is typically the next 2 solutions for the Helmholtz equation. If I combine those 2, I can map the entire cortex into a spherical shell. People currently do mapping into spherical surfaces, but can do that to a shell. So for the dipolar modes, we can calculate 3 coordinates here, and that maps you to a theta and phi. And for the radial mode, we can see how you are in terms of interpolation between the inner and outer temperature, and gives you a radial coordinate. So each point in each cortex can be mapped into a point in this hollow sphere. And it allows us to do some really interesting comparisons. For instance, comparing how big the frontal lobe is in humans and chimpanzees when mapped to the sphere, or comparing how certain parts of the brain shrink under certain conditions, or how different proportions of different parts of the brain exist for different sensory modes. The sky's the limit. Like I said, that's not something I even have done yet. That's something planted them. So this spectral embedding, volumetric spectral embedding, I think is one of the really exciting things we're going to be doing in the future. Okay, I'm going to finish just thanking my lab mates, because a lot of this presentation, not talk about the work, has been done with their help. We now have this really nice space for our lab. You're all welcome to visit anytime. It's the one nice place in the entire campus of Fundão. And I say that my students, they're not particularly well paid, but they're very well fed. I cook for them. I have these lab meetings. And again, if you're there, we're all welcome to join. So thank you very much, and I'm done for now.
Just because of time, we will take one question, but you can join Bruno for lunch afterwards. So, any questions for now? Urgent ones? Okay, it seems everyone just wants to eat, I guess. So, what? No, we should just say some communication. Yeah, I have some stuff to say. So, okay, before you go, a couple of things. After lunch at 2 p.m., we have the tutorial, so you can meet Tiago here, and then he will take you to the computer lab, okay? Second, on Wednesday at